Have you wondered how your body fights infections and heals itself every day? The answer lies in white blood cells called leukocytes. Leukocytes patrol the body identifying pathogens, removing debris, and coordinating repair. They are essential defenders and communicators of immunity. All leukocytes originate from multipotent hematopoietic stem cells located in bone marrow. These stem cells can become many different blood cell types. As stem cells commit, they produce three major leukocyte groups: granulocytes, monocytes, and lymphocytes. Each follows a distinct developmental pathway. Throughout this video, we use simple labeled diagrams and color coding to follow each lineage. Focus on structure, stages, and basic functions. Let us begin at the source and follow cells from stem cell to mature leukocyte, starting with the stem cell niche and early commitment. Hematopoietic stem cells are multipotent and self-renewing. They balance producing new stem cells and generating progenitors that will differentiate. Two main progenitor types arise: common myeloid progenitors and common lymphoid progenitors. These direct cells toward different immune families. Common myeloid progenitors generate granulocytes and monocytes, while common lymphoid progenitors produce B cells, T cells, and natural killer cells. Bone marrow signals, growth factors, and transcription factors guide fate decisions. Local microenvironment strongly influences lineage commitment. Cell morphology changes during maturation. Precursors acquire granules, shape changes, and surface markers that define their lineage and readiness. With that foundation, we now follow the myeloid branch to examine granulite development in detail. Granulocytes include neutrophils, eosinophils, and basophils. They are fast responders with visible cytoplasmic granules used for defense. Granulocyte maturation sequence proceeds from myeloblast to promyelocyte, myelocyte, metamyelocyte, band form, and finally mature segmented granulocyte. Neutrophils are short-lived but abundant. They phagocytose pathogens, release antimicrobial granules, and form nets to trap microbes. Eosinophils target parasites and modulate allergic responses. Basophils release histamine and mediate vascular changes and inflammation. Bone marrow stores and releases granulocytes on demand. Emergency signals accelerate production in response to infection or stress. That completes the granulocyte overview. Next, we will follow monocyte development and their role as tissue defenders and remodelers. Monocytes arise from the myeloid progenitor and circulate in blood as precursors before entering tissues to become macrophages or dendritic cells. Bone marrow produces monocyte precursors which mature into blood monocytes. These display surface markers that distinguish them from granulocytes. Once in tissues, monocytes differentiate into macrophages that phagocytose debris, present antigens, and secrete repair signals to coordinate healing. Macrophages are tissue-specific. Liver Kupffer cells, brain microglia, and lung macrophages show specialized adaptations to their environment. Monocytes and macrophages also direct chronic inflammation or tissue remodeling depending on signals, balancing cleanup with repair. With monocytes covered, we now explore the lymphoid lineage including B cells, T cells, and natural killer cells and their maturation sites. Lymphocytes derive from common lymphoid progenitors and include B cells, T cells, and natural killer cells, each with distinct development and roles. B cells mature in the bone marrow, then can become plasma cells that secrete antibodies to neutralize pathogens and tag them for destruction. T cells migrate to the thymus for maturation and undergo positive and negative selection to ensure self-tolerance and specific antigen recognition. Natural killer cells develop to provide rapid innate responses against infected or transformed cells without prior sensitization. Quick recap. Multipotent stem cells in marrow give rise to myeloid and lymphoid progenitors, which mature into granulocytes, monocytes, and lymphocytes with distinct roles. Understanding leukocyte maturation clarifies how immunity is produced and maintained. Review these stages to strengthen your foundational knowledge in hematology. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology, and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.